All right, imagine this for a second. You're a doctor, right? And you've got a patient in front of you. They've got the suspected bacterial infection, but you need to figure out exactly what's causing it to make sure they get the right treatment. That's where this really clever tool comes in the ONPG test.
Yeah, you're right. It's absolutely crucial in those situations.
So in today's deep dive, we're going all in on this ONPG test, how it helps scientists unlock these kind of hidden secrets about how bacteria consume sugar. We're going to be looking at excerpts from onpgtest.pdf, which is a scientific document. We'll see how this test that seems so simple on the surface actually tells us a whole lot. So, like, in the simplest way you can, what is this ONPG test really telling us?
So basically, it figures out whether a specific bacteria can produce this enzyme called β-galactosidase, which is like, a tiny little machine inside the bacteria. And this enzyme is super important because it's what breaks down lactose, a sugar in milk, into these simpler sugars, glucose and galactose. And it's these simpler sugars that bacteria can actually use for energy.
So it's like checking if a bacterium has the right tool in its little toolkit to be able to digest lactose. Pretty neat.
Exactly.
But why does any of that even matter? Like, why do we care so much about whether some bacterium has a sweet tooth for lactose?
Well, that's where things get really interesting. This tiny detail is actually a big deal for a bunch of reasons. So first off, it lets scientists figure out the different types of bacteria. Think about it. Imagine trying to tell apart hundreds of these tiny, practically identical bacteria. Knowing what they can and can't digest helps us categorize them. It's like giving them name tags based on their favorite foods. And going back to our doctor with that patient who has the infection, knowing if the bacteria causing the problem can digest lactose is going to be super important for deciding on the right treatment.
So it's like we're profiling the bacteria, but instead of fingerprints, we're using sugar.
Exactly. But it's not just about identification. It's also super important in food science, especially for things like making yogurt and cheese. Because believe it or not all bacteria are good at making yummy dairy products.
Wait, so some bacteria could, like, turn my yogurt into something gross?
Well, maybe not totally gross, but let's just say they might not give you the flavors you want. Or even worse, they could be harmful. The ONPG test helps make sure that the bacteria being used are the good kind, like certain lactobacillus species. Those are the ones that break down the lactose and make yogurt taste all tangy and delicious.
So the next time I'm enjoying some yogurt, I'll be thanking the ONPG test and all those little lactose-loving bacteria hard at work.
For sure. It's all connected from those tiny enzymes to the food we eat.
Okay, so this ONPG test, it can tell us a lot, but how does it actually work?
Yeah.
Our document mentions this thing, ONPG, and it's like a lactose imposter. I'm kind of lost.
Yeah, that's a really good way to put it. Scientists have found this really clever way to basically trick bacteria, and it all comes down to this ONPG molecule.
Okay, now you've got me really curious. Tell me more about this lactose imposter.
So ONPG, it's structurally really similar to lactose, almost like a chemical twin or something. So in the lab, when scientists introduce it to bacteria, the bacteria think, ooh, a tasty treat. But here's the thing. When the ONPG is broken down by that β-galactosidase, it releases this substance called o-nitrophenol.
Okay, I feel like there's a but coming here.
Okay.
And I bet it's a colorful one.
You got it. O-nitrophenol, it's bright yellow.
Yeah.
So picture this. You've got a test tube, right, with our bacteria suspects in it. We add the ONPG, and if those bacteria start producing galactosidase to break it down, boom, the whole solution turns yellow, like a signal flare.
No way. So they're literally telling on themselves with this color-changing reaction. That's brilliant. It's like a science fair volcano, but instead of lava, it's like lactose love.
Exactly. It's a simple visual cue, but it tells us about this really complex process. And I think it's really cool that this technique shows just how specific enzymes are.
Okay, remind me again how enzymes work. They're like these little machines, right?
Yeah.
With very specific jobs.
Exactly. It's like a lock and key. Only the right key, which is the enzyme, can open a specific lock, in this case, lactose, or that ONPG imposter. So if the bacteria, if they have the β-galactosidase enzyme, they can break down the ONPG, and we get that yellow o-nitrophenol. No enzyme, no color change.
Ah, okay. That makes sense. It's amazing how scientists can use these really specific interactions to learn so much about, like, the microscopic world.
Yeah.
Okay, so now I really want to know, how does this test actually happen in the lab? Can you walk me through it?
Absolutely. So we start by preparing what we call a broth, kind of like a soup that bacteria really like to grow in.
So we're giving our bacteria a nice little spa day.
Haha, yeah, something like that. So we put our ONPG treat in the broth, along with the bacteria that we want to test. And we have to be really, really careful to keep everything sterile. We don't want any other bacteria crashing the party.
Right, like those people who show up to a party uninvited and eat all the snacks. Not in our bacteria spa. We need those results to be accurate.
For sure. So we use special techniques and tools like sterilized loop. It's like a really tiny spoon to move our bacteria without contaminating anything. So once the bacteria are in time to do their thing, so we keep them at a comfy temperature, like body temperature, for a day or two.
So it's like a little incubation vacation for the bacteria. And if they have the right enzyme, they'll break down that ONPG and the solution turns yellow.
You got it. It's all about what we see after that incubation time. If the broth is still clear, no galactosidase. But if it's that bright yellow...
We know those bacteria are having a blast with that lactose imposter.
Exactly. That color change is confirmation. It's how we know the bacteria actually have the enzyme.
So simple, but so smart. I love how it's this mix of basic chemistry and really clever observation.
It just goes to show how scientists can use things we can actually see to understand things we can't, like bacteria and enzymes. And to kind of illustrate that, our document actually gives us some examples of this test in action, which I think your listeners will find interesting.
Definitely. Let's hear about those bacteriolases. Okay, so we've got our color-changing test. We're like bacteria detectives who are we analyzing in our ONPG lineup.
Well, the document points out a couple of familiar names. First up, Escherichia coli, or E. coli, as most people know it.
Ah, E. coli. I've definitely heard of that one, but I pretty much just know to, like, not get it in my food.
Yeah, that's a good rule of thumb, generally speaking. But E. coli is actually found naturally in the intestines of both humans and animals. And there are a lot of different strains. Some are harmless, some not so much.
Okay, so that's where the ONPG test comes in, right?
Yeah.
To figure out what we're dealing with, the harmless kind or the kind that crashes the party and makes everyone sick.
Exactly. And in the case of our E. coli suspect, we get a nice bright yellow result with the ONPG test.
A lactose lover. So E. coli is ONPG positive.
Exactly. It produces that β-galactosidase enzyme, breaks down the ONPG, and that color change tells us what's up. But not all bacteria are so keen on lactose.
Interesting. Okay, who else is in our lineup? What other bacteria are we putting under the ONPG microscope today?
The document also mentions Proteus vulgaris, another common bacterium, often found in things like soil and water, and even in the human gut.
Okay, Proteus vulgaris. What's its deal?
Well, unlike our friend E. coli, this one is ONPG negative. So we wouldn't see that bright yellow color because Proteus vulgaris just doesn't have the ability to make that β-galactosidase enzyme. It's not equipped to break down lactose, or our clever little ONPG imposter.
That's wild. It's amazing how two bacteria can be so different when it comes to what they like to, eat.
Yeah.
And this simple test can just show us that difference, like a personality test for bacteria.
I like that. It really shows how diverse and complex the microbial world is. We think of bacteria as these simple organisms, but they have all these amazing adaptations and abilities, and it's so important to understand these little details.
It's like we've peeked behind the curtain of like a whole hidden universe.
Yeah.
Okay, so as we wrap up this deep dive into the ONPG test, what are the big takeaways for our listeners?
Well, I think the most important one is that this test, this really clever test, gives us a glimpse into how bacteria work their metabolism, which we can't actually see. And just by figuring out how they process something as simple as sugar, we can learn so much about them, how to tell them apart, whether they might cause problems, even their role in making delicious food.
It really shows how everything is connected, right? From these tiny enzymes working away inside these bacteria to the yogurt we have for breakfast.
Absolutely. And it also shows how creative scientists are developing these amazing tools. The ONPG test really shows how curious we are as humans, this drive to understand everything around us, even if we need a microscope to see it.
It's true. We started with a simple question, can this bacteria digest lactose? And we ended up with this really elegant, super insightful test. On that note, is there like a thought-provoking question our listeners can think about as they go about their day?
Here's something to consider. We've seen how a simple color change can reveal this whole process happening in bacteria, the enzymatic reaction. What other hidden processes, maybe even in our own bodies, could we see if we had similar tests?
Wow, that's a great question. It makes you think about everything that's happening inside of us all the time, even if we don't realize it. Well, that's our deep dive on the amazing ONPG test. Be sure to follow us so you don't miss our next adventure into the world of science. Until then, happy exploring.